Peace be upon you all, and welcome to this new exciting session with Dr. Fuz Rana. And today we are going to have this new installment of Seeing God in Biology, and this will be the last one in the Central Dogma, wouldn't it be, Fuz?
Yes, we've taken quite a bit of time working our way through different facets of the central dogma, but for good reason, because this is the organizing principle in biology. And so we are finally reached the end of the flow of information, at
Yeah,
least with respect to the central dogma. But there's still many more topics that we want to discuss when it comes to seeing God in biology. So
absolutely,
this isn't the
absolutely.
end, it's just the end of the central dogma.
So we're gonna be looking into translation. So translation is like we're moving now from RNA to proteins, right?
Yes. Yes, and so here's a quick reminder of what the central dogma is, and it
Okay.
essentially uh, is
Whoops.
the way in which information flows, in biochemical systems, and you know the um, uh, the the information that the cell needs to make its component parts and to carry out its operations are found within the structure of DNA.
Mm-hmm.
And then that information is copied through a process called transcription, where uh, for uh, for proteins, that information is in the form of messenger RNA that makes its way to ribosomes, where that information is read and translated from the language of nucleic acids, of DNA and RNA, into the language of amino acid sequences or into proteins. And so that's what we're going to be talking about today. And, as we've seen throughout the process, there is a logic to the central dogma. There is this elegance and sophistication to the process of DNA replication and transcription, where we see God's fingerprints so clearly evident in these processes. And what we'll see today is the same thing, that there's remarkable evidence for design in the process in which proteins are made using the messenger RNA as the code source to direct the production of proteins.
Okay, so let's see how we get into the machines, yeah, the proteins.
Right. And so the heart of protein production, of protein synthesis in the cell, is essentially a particle that is found inside the cell called a ribosome. And these are three depictions of ribosomes that you might see in a biology textbook or
Mm-hmm.
elsewhere. On the left is a simplified structure for the ribosome, which consists of two subunit parts that are cleverly named the large and the small subunit. So biochemists
Mm-hmm.
are very creative in how they name things. And
Yeah.
then the middle structure is the same ribosome, but now what we're seeing here are what's called a space filling model, where we see the contour of the ribosome with respect to the structures that are formed from the individual atoms as they
Mm-hmm.
interact. And then the on the right, that's called a ribbon diagram, where it shows the backbone of the proteins and the RNA molecules that make up ribosomes. Ribosomes are massive complexes made up of, depending on whether it's a eukaryotic or a prokaryotic ribosome, probably somewhere between 80 to 100 different proteins, as
Wow.
well as as well as three very large RNA molecules that are called ribosomal RNA molecules. So these are massive complexes, and they make up a significant portion of the biomass that are found in cells. So in bacteria, a typical bacterium, there's somewhere between 10 to 20,000 ribosomes in a single cell, and ribosomes make up about a quarter of the weight, or the quarter of the mass of a bacterial cell. So these are
Wow.
uh, essentially the site of protein synthesis, where the protein is produced and where the messenger RNA is read. So in eukaryotic cells, ribosomes can exist in two populations.
I can only restate the comment that the machine that will now convert RNA into proteins is itself made of proteins. So
Yes,
one more paradox. Yeah.
right.
Yeah.
It is. Yeah, it's the queen mother of all chicken and egg
Yeah.
paradoxes.
Yeah.
Yes.
And there aren't any ribosome variant that is purely RNA. It
No.
must have proteins. Actually, the majority of it is proteins, as you've just explained.
Yes.
All right.
Most of the mass is probably protein. Yes.
Wow.
But now in eukaryotic cells with a nucleus, there are two populations of ribosomes. One are called cytosolic ribosomes. These are found free in the cytoplasm. And then the other are associated intimately with structures that are with a network of membranes called the endoplasmic reticulum. And the region where the ribosomes are located is called the rough endoplasmic reticulum because it has that rough surface appearance because of the ribosomes. But again, the whole point of this is to really emphasize the fact that it's not just 2 or 3 ribosomes inside the cell. These things are one of the major cellular components, whether it's prokaryotic cells or eukaryotic cells. And these are, and there are different populations of ribosomes in eukaryotic cells that actually are producing different types of proteins and are helping to deliver the proteins to particular compartments in the cell that is critical for the function of that individual protein. So the ribosomes are, again, distributed everywhere inside the cells and are playing really very important roles.
Okay.
This is a cartoon showing the structure of the large and the small subunit, or at least the composition of the large and the small subunit. And on the top, we see essentially the ribosomal RNAs. The red ribbons are representing the ribosomal RNAs. And in in bacteria, in prokaryotic cells, the small subunit consists of a single, very large ribosomal RNA and about 30 different and distinct proteins. And then the large subunit has 2 RNA molecules, one very large, one smaller, and then about 50 proteins. And then the 2 subunits will come together to form the ribosome proper. And they typically will come together where the interface between the 2 subunits is the region where the messenger RNA produced in transcription will actually interact with the ribosome. So that interfacial region is where that messenger RNA is located.
Wow. So it's designed to like clamp on it from up and below, and then it will pass to be processed.
Yes.
Sort of one more feels to me like one more Turing machine there.
Yes. Yeah. And
with an input tape and an output from the other side.
Yes. There is definitely ribosomes really are very much, like the type of manufacturing machines you would see in a plant, but also they are, real life physical examples at a molecular scale of an abstract machine that Turing invents as part of computer science. So yes, very much Turing machines. Now, the difference between the ribosomes in eukaryotic cells is the size of the subunits. Both the small and the large subunit are a little bit larger. I'm drawing a blank now on the number of RNA molecules in the large subunit of eukaryotic cells, but the number of proteins is larger. You typically see close to about 100 proteins or so combined in the small and the large subunit. So very similar structures. It's just that you see more protein components in the eukaryotic ribosome. I think it's still one ribosomal RNA molecule in the small subunit, and I think it's still 2 RNA molecules in the large subunit, but just the number of proteins is different. And there's actually
one more example of having like the same concept, but with different constructs, with different designs that lead to the same function.
Yeah, and probably what we see happening in eukaryotic cells that I don't believe happens in prokaryotic cells is that there is a core set of proteins in each of the small and the large subunit, but then there are actually distinct populations that are independent of the two, cytosolic and rough and ribosomes that are associated with the endoplasmic reticulum. There are also subpopulations within the cytosol and within the attached to the ER that have variable protein structures, and those variable protein structures make those particular ribosomes distinct in terms of the production of certain types of proteins inside the cell. So it would be similar to, thinking about manufacturing. While there are a number of different assembly line processes that exist to manufacture a wide range of products, and those assembly lines may have core designs, they also have unique aspects that are tailored to the particular product that's being produced. So it's a similar concept that you see with regard to ribosomes in eukaryotic cells, is that there are distinct populations.
Amazing. Yeah. All right. Okay. Great.
Okay.
So I feel also sometimes like ribosomes are like those, I don't know, 3D printers or something. So or CNC machines. So they're just getting the instructions and producing the output thing. Now, how this output thing shapes into a 3D structure, the folding of the proteins. We are going to address the folding of the proteins today, are we?
That'll be, I think, a separate episode.
It's a later episode. Yeah. Correct.
Yeah. So I think, in fact, the next episode will be on protein folding.
Yeah. Yeah, absolutely right. Yeah.
Now this slide is inspired by you, Ahmed, because you
the why.
you love to ask the why questions. And again, as we've mentioned before, there's something really very profound about asking why, right? And it goes beyond just simply an idle curiosity, because, as you've pointed out, the why question really suggests purpose in
absolutely
in meaning. So,
all right. So thank you for preemptively answering it.
Yeah, sorry about that.
No, that's absolutely.
I need more work as a
fantastic
host for a show. But in my book Fit for a Purpose, I have a whole section in the book where I talk about why ribosomes are the way they are. And there are some biochemists that are curious, that will ask the why question. And these are some of the why questions that are asked. For example, why are ribosomes made up of so many proteins? Right? Why are ribosomal proteins all the same size, and why are they smaller than typical proteins? So this is something that's really interesting, is that the ribosomal proteins tend to be smaller than typical proteins, and they almost all are of the same size. And then why are ribosomes made up of so many proteins but so few RNA molecules? And why are the RNA molecules so large? These molecules are huge molecules. And then why do ribosomes employ ribosomal RNA as the catalytic subunit instead of proteins? So most enzymes are made up of proteins. There are some enzymes made up of RNA. These are called ribozymes, but most of them are made up of proteins. And so the question is, well, why wouldn't we expect to see, the catalytic portion of the ribosome being proteins? Why wouldn't we expect that? But instead, what we see is the opposite, which is it's actually RNA that's performing the catalytic function. So ribosomes are ribozymes. And, I remember being at an Origin of Life conference years ago when the late Leslie Orgel, who died a few years ago now, who was a prominent Origin of Life researcher, said, "I'm absolutely convinced that the RNA world must be true because ribosomes are ribozymes. To him, that was a huge insight and so the argument is, well, ribosomes are, because the ribozyme because the RNA is the catalytic part of the molecule, then it means it must have been in existence in the RNA world, and the ribosome is this leftover remnant of the transition from the RNA world to the DNA protein world. And so, he was arguing that, that, and others, that the structure of the ribosome has no real rhyme or reason. It's just what evolution haphazardly produced.
Wow.
But what we're going to see with these why questions is that there's actually reasons, and those reasons really undermine, I think, the RNA world hypothesis. And so,
Wow.
The reason why, let's see here, the reason why ribosomes are the way that they are, why, for example, they are made up of proteins that are of the same size and that are smaller than most proteins. It can be found in the recognition that before a cell reproduces, before it replicates, the cell's machinery has to produce essentially a duplicate set of ribosomes, so that when the daughter cells form, each of the daughter cells forms with more or less the appropriate number of ribosomes.
Yeah.
Right. That makes sense, because you can't have a daughter cell that is unable to make proteins at an efficient rate. Otherwise, it's not going to survive. And again, this is a massive undertaking because there are 20,000 ribosomes that are found in a typical bacterium. So this is a huge undertaking. Well, it turns out that the most efficient way to make a ribosome is to make it with proteins that are smaller than a typical protein, because that allows you to make those proteins as fast as possible, right? The larger the protein, the longer it's going to take. So if you make up if you have the same biomass of proteins that's distributed among a larger number of individual proteins, that actually allows you to make the protein component more rapidly, because they can all be made simultaneously. And if they're uniform size, it means that all that full set of proteins will all be available at the same time. If they were variable in size, the small ones would be waiting around for the larger ones to be produced, and you would wind up with compositional mismatches.
Wow.
So the protein biomass has to be distributed in a large number of proteins that are, again, of uniform size and are relatively small. But if you have too many proteins, or if the proteins are too small, that actually compromises the efficiency of the process, because now all the initiation steps become the rate limiting
a large overhead. They become a large
overhead. And if you and if you're having to make too many, that defeats the purpose. So there's an optimal number of proteins in ribosomes of an optimal size and of an optimal size distribution. Now, it turns out that ribosomal RNAs, because they are just simply transcribed, they don't have to go through the process of transcription and translation. There, it's more efficient to make very large RNA molecules.
Because it's just a single step.
It's a single step. And so it's actually faster to do it that way. And then also, as we're going to see in a minute, the integrity of the ribosomal protein is very important. And to ensure that you are producing ribosomal proteins with a high degree of likelihood, a high degree of fidelity, a low likelihood of error, means that you also want to have relatively small proteins as well. So there's actually a rationale for, and it turns out that eukaryotic cells, in prokaryotic cells, which reproduce very rapidly, you're better off having a a smaller proportion of proteins relative to RNAs. If you're in cells that reproduce at a slow rate, then you can actually have more proteins relative to RNA, and we actually see that, which explains why there's a difference in the number of proteins that we see in eukaryotic ribosomes versus prokaryotic ribosomes. And it turns out that in mitochondria, which can reproduce at a fairly slow rate, and mitochondria have ribosomes in them as well, there's a very large number of proteins compared to the RNA molecules because the rate of replication is really slow. So you actually see this logic reflected in the compositional makeup of ribosomes.
So there is a pattern. There is a pattern that confirms the hypothesis, actually.
Yes, exactly. And again, as I mentioned earlier, having a larger number of proteins in eukaryotic ribosomes gives you the opportunity now to create compositional variability and create subpopulations that can be highly specific. So the whole point here is that there's an elegant rationale for why again ribosomes look the way that they look. It's not haphazard whatsoever, but there's again an elegant rationale for why they are the way that they are.
Wow. And, actually, it just reminds me also of a little bit of analogy in programming, and so on the macro level, the trend today is to do programming in what is called agile methodology, where you will not just go very big chunks of development or stages of development. You'll do little ones of them. Each one of them would produce an output, and you call them sprints. So this is today the pinnacle of the human thinking for how to produce large-scale projects on small chunks. Each one will be successful on its own, and then go incremental. And on the other hand, the whole point when you go to the micro level of parallel processing, okay, in the old days, you'd have one processor and you'll try to make that processor as strong as possible, and it's just doing things in series. But modern computers will have multiple cores, and you can also have computers that are working in parallel processing. And what the operating system will do to exploit that is to split one task into many chunks that can now be run in parallel, so that sophisticated computations can actually be split into small chunks. And then you have the same overhead issue that you're talking about. If the chunks are too small, then the initiation and termination and passing over of the intermediate results will be too much of an overhead. So you need to reach to an optimal size of how you chop it, because if you chop it too small, it doesn't get efficient. If you make it too large, then you're not making any benefit from the parallel processing. So it's just mind-boggling and mind-blowing that essentially the same concepts of how parallel processing should work and how agile development, actually, there is a combination of both into what you have just described. And once again, this can only happen if there is real supreme sophisticated intellect behind all of this. So I hope that the analogy it really clicks, and people who are hearing this, especially those who are software and electronics engineers, will get the gist of it. What do you think? How do you think?
I think that's beautiful, because the point you're making is that there's a set of concepts that are used today in programming that have an undergirding logic that is all built around efficiency, right?
Yeah.
And this is and the same concepts are manifested, in the design in the compositional makeup of ribosomes. And ribosomes are incredibly complex systems that we still don't have a full understanding of by any means, right? And so, but what's interesting is that when we start asking the why question, we see a rationale that undergirds it, and it's very encouraging to me to see, because as a biochemist, that rationale intuitively makes sense. But then to see that rationale is actually integral to the design of information systems, which is what the ribosome is yeah, as you said, it's mind-boggling. This is the type of thing that
Yeah.
keeps me awake at night,
And
thinking about what this means.
Essentially, because the direct counterpart of what the ribosome is. So we have the RNA, which is now a program in memory, and what the ribosome is essentially it's like a processor. It's like a core. It's taking this program that's now in memory, it's now interpreted or compiled after splicing, and it's executing the instructions there to produce the output, which is the protein that's gonna go eventually to a 3D printer to be folded or something later. But so this is a processor. And guess what? In the problem that you are describing, where you have a sophisticated machine, which is now the ribosome itself, but I guess this is also applied to other sophisticated machines, like I don't know, flagella or other stuff, where you have so many components, and then there are all those little proteins that come together in intricate designs, and it just lends itself to a parallel processing paradigm where we need to produce so many ribosomes, so let's have them split up into those little chunks so that we can parallel process them. Interestingly enough, using other ribosomes.
Right.
So the whole thing is just a big loop. And again, it is again another example of this, having end in mind and having a foresight, where the inputs foresee the outputs, and the whole system is beautifully integrated. So, wow, what a great insight to tell us that the why question here has really very nice answers too.
Yeah.
Yeah, and it remains the question of the ribozyme versus the protein for the catalytic action. Do we actually have an answer for that too?
Yeah, well, because the ribozyme is actually, catalytically speaking, less efficient than if you were using a protein catalyst, and I believe the answer is actually connected to the fact that the ribozyme can be produced in a single shot, right? Mm-hmm. I can't remember which of the ribosomal RNAs is actually possessing the catalytic component to it, but I know that the 16S has the sequence that aligns the messenger RNA, but I can't remember which one is the catalytic unit. So off the top of my head, but again, it has to do with, I think, efficiency of production. Is that you can make the ribozyme very quickly, in a single shot.
Yeah.
And so you're less likely to introduce, I think, error into that structure, because you're making it,
right away,
as a single step. That it's not two steps, right? Where it's got to be transcription and then translation, where each one could potentially introduce error.
Right? All right. So what do we have here? This is the structure of a ribosome, I guess.
Yes, and so this is we're starting to drill down into a little bit more of the details of the process, because we talk about, again, the RNA molecule being positioned at the interface between the large and the small subunit, and it's directing the production of proteins. But then the question becomes, how do those amino acids that are used to build proteins brought to the ribosome? So there are molecules called transfer RNA molecules that are binding the individual amino acids, and there are specific transfer RNA molecules that correspond to each of the amino acids. They bind them, and then based on the instructions in the messenger RNA, they will essentially have the right amino acid or right transfer RNA positioned with the right amino acid into one of three sites called the A site, the P site, and then the E site. And we'll kind of review in a minute what is happening in each of those individual sites. But the transfer RNA molecules are what are bringing the amino acids to the ribosome for the process of protein chain assembly. Now, as we mentioned in an earlier episode when we introduced the central dogma, what and we'll have a whole episode on the genetic code in a couple of episodes. There's a set of rules that relates the information in DNA to the information in proteins, and this set of rules is called the genetic code, where there's what's called a codon or a coding triplet, combinations of 3 nucleotides that specify each of the amino acids. And so in the transfer RNA, there is what's called an anticodon that recognizes the codon in the messenger RNA, and that is how, through that molecular recognition process, how the right amino acid gets into the right position during the synthesis process. This is how the instructions are executed. It's through the genetic code, which is physically instantiated in the transfer RNA molecules.
So the transfer RNAs are those light blue things carrying the little beads, which
Right.
are essentially one of 20 amino acids, and then they go in through the A site, right?
Yes.
Yeah.
Yeah.
Right.
You have 2 sites, A and P, and it's we'll kind of get into what those sites are doing in a minute. But
Right.
What's striking to me again is just this, and you've already brought up this point, is just the similarity of this to a Turing machine. And so we don't have to belabor this point anymore, other than to say that this is literally a machine, an information processing machine that is converting information in one format into another in this case, where the input would be the messenger RNA sequence and the output would be actually an amino acid sequence that's making the proteins. So this is a very sophisticated Turing machine that is actually changing formats. It's still digital information, but the format is being changed as part of the Turing machine function, which is, even as a biochemist, that naively, that seems to be pretty crazy to me.
Absolutely. That's completely amazing.
Okay, so let's talk about the transfer RNA molecules. So this is what they look like. So a transfer RNA molecule is about 80 genetic letters or 80 nucleotides in length, and the chain will fold on itself, so it can actually undergo hydrogen bonding interactions with itself to form 3 loop regions. And so this is called the clover leaf design, because there are 3 loop regions. And the 2 regions that we want to pay attention to is the one of the loop regions that contains an anticodon. And then the other, it's not really a loop region, but it's called a stem. So there's a stem and 3 loop regions, just like on a clover leaf. There's the stem and the 3 clover leaves. And that stem is where the amino acid binds. And then the what are called the D loop and the other loop are loops that are regions of the RNA that are interacting with an enzyme called aminoacyl synthetase, or it's sometimes called an activating enzyme or a charging enzyme, which is the enzyme that attaches the amino acid to the transfer RNA. And if you take that 2-dimensional RNA structure and you kind of twist it, you actually wind up with an L-shaped geometry where the anticodon loop is at one end of the L, and then the amino acid is at the other end. So it's you kind of have a hook design, right? So the anticodon is interacting with the messenger RNA, and then the amino acid is then dangling. So it's putting it in the right position in 3-dimensional space, so that the protein chain synthesis can take place.
Wow. Just the very idea of this single. So the tRNA is essentially a single strand of RNA that essentially reattaches to itself to form this 3D structure, to the extent that at the bottom will be the part that actually plugs into the mRNA,
Yes.
and hence corresponds to it up there at the stem, the amino acid. And then when they go in sequence, then amino acids will be sorted according to what's on the mRNA tape, right?
Yes.
This
Yes.
is what it does. And
Yes.
it's just completely striking that you would have this single strand that is so beautifully engineered in the way that it will just, number one, just like fold into the proper 3D structure,
Right.
and number two, have all these attachment mechanisms to other enzymes to make the amino acid attach. And on the other side, it's exactly what you need to attach to mRNA. Again, irreducible complexity and integrated system, foresight, having end in mind, all just in one thing. And again, the paradox of RNA translating RNA, both
Yeah.
need to come from DNA one way or the other.
Yes.
Yeah.
Well, and there are, again, transfer RNAs that correspond to each amino acid, and what defines that transfer RNA is essentially the anticodon loop, right? That's what defines the transfer RNA molecule. And so
Yeah,
this
so
is
the transfer RNA is also what does the mapping between
yes,
the 3 nucleotide set, which is essentially a codon,
yeah,
and the proper amino acid. So this is
right,
the translator, the read translator
right,
guy here.
yeah. So if you think of the structure of, again, transfer RNA as being essentially it's the instantiation of the code. So the anticodon loop is the nucleic acid language, and then the amino acid is attached again to the stem, and that attachment, so the transfer RNA is essentially the adapter. It's the structure that is and when you think of the L shape, it starts with one language, and at the end of the molecule is the other language.
It's the
right?
other language. Yeah. Actually, we call this in programming mapping tables. So this is essentially an implementation to a mapping table where you have at one end language A or code set A, and at the other end code set B, and the table is what actually connects the dots. And it is totally amazing that this anticodon slash amino acid thing is manifested in a chemical structure that looks like this. So it's completely mind blowing. And those enzymes that attach the amino acids are protein enzymes, or are what kind of?
Yes. Well, the enzymes that attach the amino acid to transfer RNA are called amino acyl tRNA synthases, and or sometimes they're called activating enzymes or charging enzymes. And this is maybe a little bit more elaborate than needs to be, but it's an energy-driven process where the amino acid in a molecule of ATP, which is a fuel molecule in the cell, interact with the enzyme to form a complex where the amino acid is attached to the AMP component portion of the ATP molecule, and then a transfer RNA molecule can then interact, and then there's a transfer of the amino acid from the AMP to the transfer RNA molecule. So that's essentially the process. And again, there's corresponding charging enzymes to each transfer RNA molecule.
Wow. And the synthase itself is a protein, or what kind of molecule?
Yeah,
it's a protein.
It's a protein. Yes.
So once again, tRNAs themselves will not work without having proteins already in the picture.
Yes, exactly.
And each one of them needs. So you have amino acyl tRNA synthase that is amino acid dependent, or each amino acid needs its own synthase. Did I understand you correctly?
Yes.
So you need a set of 20 protein enzymes already existing for tRNA to make its job.
Yes.
So one more chicken and egg paradox.
Yes, and in fact, the minimum number is 20, I think there's actually about 30. There's some tRNA and amino acid pairs that actually, there's some amino acids that are recognized, that are attached to two different types of transfer RNA molecules. And this gets into the redundancy of the genetic code, which we'll pick up in a couple episodes. Yeah. So
wow.
Okay.
All right. So amazing.
Yes. And so now here is again another depiction of a ribosome and the messenger RNA. And what I'm going to call your attention to is in the middle of this diagram, the three the three colors, kind of the reddish color, green and yellow, and these are essentially the A, P, and exits, E sites, or the exit sites.
Okay.
So these are very important sites within the ribosome. And so here's another more simplistic cartoon showing the ribosome. And remember, we said a couple of episodes ago that with messenger RNA, there is an untranslated 5' region, right? That is part of the code, and that is what actually interacts with one of the ribosomal RNAs, I think the 16S in prokaryotic cells. And that interaction aligns or positions precisely the messenger RNA on the ribosome. So that interaction is really very, very critical.
Because if you don't have the proper alignment, then a shift in the codons will mess everything up, right?
Right.
You need to start exactly in the right place, because
Right.
all the three nucleotide triplets,
Right.
if you shift it, you ruin it, right?
Right. And then remember we said, like with DNA replication goes in the 5', 3' direction. In RNA, the synthesis is 5', 3'. At the ribosome, the reading of the messenger RNA is 5', 3'. So it's read in the opposite direction. But that makes sense because in prokaryotic cells, once transcription starts, translation begins immediately. So even before the RNA is completely transcribed, the cell's already beginning to translate it. And in fact, many times you'll have multiple ribosomes translating the same piece of RNA at the same time. And so the directionality is actually is part of the design to allow in prokaryotic cells, very efficient protein production. Even before the process of transcription is done, you might have several ribosomes reading that RNA right away. And of course, this allows for a very efficient, very rapid process. Now, in eukaryotic cells, transcription and translation are separated in space and time, but not so in prokaryotic cells.
So in prokaryotic cells, one end of the RNA is still being produced and the other end is already being read. So it's very fast.
Yes, extremely fast.
And unbelievable. No wonder bacteria just reacts super crazily fast to everything. We can't kill those guys.
Yeah,
we can't get rid of them. Okay.
So then I'm going to try to talk through this. Let's see if I can. But remember, we have the A and the P site. So what happens is that the transfer RNA that corresponds to the amino acid in the very first position goes initially to the P site. And then the second position goes to the A site. So now you have two transfer RNAs with a single amino acid in the A and the P site. What ends up happening is the amino acid that's in the P site will react, mediated by the ribosomal RNA, will react with the amino acid in the that's located in the A site. Sorry, the amino acid that's attached to the transfer RNA in the A site, so that the number 1 amino acid is now attached to the number 2 amino acid. then that you have an empty a transfer RNA that is now uncharged, no longer has an amino acid. It will move to the exit site and then will leave the ribosome
and get out.
Then the amino acid that's now in the A site that has, the transfer RNA that is in the A site that has amino acid 2 and amino acid 1 attached to it will now hop over to the P site, and then the next transfer
Will come to
RNA
fill.
for number 3, and then you now get the dipeptide, the 2 amino acid residue transferred over, attached to the transfer RNA in the A site, and then that P site leaves, and that process is repeated over and over again until the entire amino acid chain is produced. So it's an assembly line type of process where you literally are ratcheting along the messenger RNA codon by codon in building the protein chain in that way. So it's
actually we can show a video for that right now.
Okay, perfect.
Yeah. Okay, I think this was a great video to explain what you've just said, and it looked really wonderful how those guys are getting in, stitching to the P, and then the one at the P ejecting, and this chain keeps growing until it's finished. It's it's like somebody making a necklace out of beads, and
yes,
keeps putting in a new bead until it's finished with all the colors there. So it has an element of beauty in it.
Oh yes, yeah. Well, and when you look at the ribosome in action, it looks like it's the analogy is a Turing machine, or but it really is a machine in every sense of the word. And it reminds me in a sense of an assembly line or of a manufacturing operation. And in fact, biochemists have discovered that there are these quality control systems that are integral to the process of protein synthesis. So what we've seen is really just the tip of the iceberg, because there are, again, these quality control checkpoints that are implemented into every step of the process of protein synthesis. Now, I before joining Reasons to Believe a couple of decades ago, I spent nearly 10 years working for a Fortune 500 company, and I worked in R&D, but we would do work from time to time with people at the manufacturing plants, and everybody complained about quality control because it always felt like it was getting in the way. But soon you came to appreciate that quality control is really very important when you're trying to produce a product, because you want the consumer to get what they think they're getting when they buy your product.
Yeah.
And there's one way you can do quality control, which is you just simply examine the product at the end of the manufacturing process, and if it's not good, you throw it away. Well, that's the worst way to do quality control, because it's so inefficient. It's better to do checks along the way before you invest more resources and energy into the process manufacturing a defective product. And the sooner you can detect an error, the greater the opportunity you have to maybe rework that step and even salvage, the what was produced up to that point.
Yeah.
And you see that same set of principles in implemented in the quality control systems for protein synthesis. And so to me, in the quality control, you even see evidence for intelligent design. It's not just in the nuts and bolts of the protein synthesis process, but it's in all the decorative aspects of the process that are very, very important in terms of ensuring quality products. And these quality control systems are absolutely non-negotiable. And the reason why is because if you don't make ribosomes appropriately, you're going to wind up the cell's going to wind up in an autodestructive cycle.
Uh-huh.
right? Because if ribosomes, which are making proteins, are made out of proteins,
Hmm.
and those proteins in turn are being used to make the ribosomal component of the ribosome. So proteins are needed. Ribosomes are made up, which make proteins, are made up of proteins. So you
So
need proteins to make.
it will be an error accumulation process that will essentially self-destruct.
That's exactly right. But the proteins are not only the ribosome themselves, but they're used to make the other component, the RNA component of ribosomes. So you can't escape it. And as you point out, if a ribosome is defectively made, then it means the proteins that it's going to produce are going to be defective, which means the ribosome that results is going to be defective, which means the
itself.
next round of proteins are going to be even more defective. And so this is another reason why ribosomes can't evolve. You can't have a crude, cumbersome ribosome that then becomes more and more sophisticated over time. If it
Uh-huh.
doesn't meet a certain level of certain standard of fidelity at the very onset, you're going to wind up in this auto-destructive cycle. You can't bootstrap a ribosome into existence.
Yeah.
That then is followed by some kind of evolutionary optimization.
I see. So what you're saying is that ribosomes must have been in a proper design from the very beginning. Otherwise, if it starts in the wrong place, it terminates. It's a dead end, and this is even more of an argument, not only an argument of irreducible complexity and integrated system, but it's an argument of either top quality or nothing. So you'd rather get it right from the very first try, or you go nowhere.
Yep.
So it has to be with its quality control, with its brilliant and magnificent design, with all the components coming together, and there's plenty of them. Otherwise, it doesn't work, and this means that it's design or nothing. It's intelligent design or nothing. Essentially, what you're saying.
Yes. Yes. And so what we can do is just spend a few minutes talking very quickly about some of the quality control points in the process. And there's, again, a rationale behind why you have quality control at these particular points. And, and so this is not just haphazardly assembled through a historically contingent process, but there's a rationale. So, for example, there are these very elaborate processes in place to make sure that transfer RNA and ribosomal RNAs are manufactured appropriately. And if these are made incorrectly, they are immediately destroyed. The cell, it has zero tolerance for any mistakes in the production of tRNAs or ribosomal RNAs. And this, of course, makes a lot of sense.
There's a mechanism that will find defective ones and will degrade them.
Right. There's an ensemble of enzymes that work together to detect the error and then to actually make sure that the RNAs are destroyed if they are made inappropriately. I don't know that I know how that the system detects faulty RNA molecules or faulty tRNA molecules, but you cannot have anything short of almost near perfection in this step of the process, because it's like producing a machine on the assembly line. That it's like you've got an assembly line for making machines that are part of the assembly line. Right?
Yeah.
If the assembly line machinery isn't made well, then you're never going to make future assembly line components appropriately. You can't tolerate error here at all.
It's downhill from that point. If you just mess up with the manufacturing equipment itself, it's straight downhill. There is no return.
Yes. And then of course, if the messenger RNA are the manufacturing instructions, you want again, those instructions to be produced with the highest degree of fidelity possible. And with regard to the RNA polymerase, there's a proofreading mechanism that is employed to make sure that the transcription process occurs with a high degree of fidelity. And then when RNA is spliced, right, this would be for eukaryotic cells. When the RNA is spliced, there are all these mechanisms in place to ensure that the RNA is spliced appropriately. If it's misspliced, it's immediately destroyed. And so there are, again, there's machinery checking to make sure that the splicing has happened appropriately. And if somehow the improperly spliced RNA escapes that mechanism, the nuclear pore, which the RNA has to pass through to go to the cytoplasm to be translated, will not allow improperly spliced RNA to pass through the pore. So there are these, again, redundant mechanisms in place to ensure that the quality, to ensure quality in the message itself. Now,
so just for the audience, the nuclear pores are those little openings in the membrane of the nucleus, and they're not just pores. What you're telling us is that they have intelligent detection mechanisms that if a product is not a proper product, it will not get out to the cytoplasm to be translated. It will just be stopped there and be degraded and call it a day. So if this thing escapes all error correction and the detection mechanisms, there is this gatekeeper at the door of the factory that will tell it, just no. You can't get out of here, because if you go out to the assembly line and become a machine there, you're going to cause us problems. So guys, this guy does not have a pass out, right?
Right.
Yeah.
Now, another place where quality control is very important is attaching the amino acid to the transfer RNA. Because if you attach the wrong amino acid to the wrong transfer RNA, you're not going to be able to produce a protein. And you probably see one of the most extreme examples of quality control at this particular point in the process. And so there is I think the error rate is about 1 in 3,000. So for every 3,000 amino acids that are attached to transfer RNAs, there is only a single error that will occur. And the mechanism is very complex, but it's all in the amino-acyl tRNA synthase, where, once the amino acid is attached, there's a proofreading mechanism that will ensure that the right amino acid is attached. And if it isn't, it'll cleave the amino acid and it'll repeat the process until the right one is attached. Wow. And then, in addition to that step, there is another step where there are these proteins, these elongation factors that will bind the amino acid transfer RNA complex and ferry it to the ribosome. And if the wrong amino acid is attached, it is this protein can recognize that the wrong amino acid is attached, and it won't bind it. It won't bring it to the ribosome. And so
amazing.
So you're seeing at every step of the process.
So if it escapes the first one, it will not be transported, and if it gets, just in case it gets transported, then it will be detected later in another stage or something from what we have described.
Right. And so, you've got, quality control checkpoints at every step of the way. Now, people have done calculations to determine what is the error rate and for making proteins, and the two benchmarks would be proteins that are made up of about 300 amino acids, and then proteins that are made up of about 1,000 amino acids. And so if you, and there's a formula that is used where the probability of producing a protein free of error is equal to 1 minus the error rate raised to the nth power, where n is the number of amino acids in the protein chain. So for a protein that's about 1,000 amino acids, which would be a larger size protein, if you had an error rate of 1 in 100, that would be 0.01, you would in effect have a probability that is so low of producing a protein that's 1,000 amino acids in length. It's so low that it in effect is a zero chance. If it's 1 in 1,000, you have about a 37% chance of making a protein that without error that's 1,000 amino acids in length. If it's 1 in 10,000, it's about a 90%. If it's 1 in 100,000, it's a 99% probability. It turns out that the error rate is 1 in 10,000. And that gives you a 90% chance of making, proteins of 1,000 amino acids correctly. So that's a very high, rate of fidelity in terms of the manufacturing operation. Of course, why not 1 in 100,000? Well, in order to get that level of accuracy, it would be so resource intensive that the rate of protein production would not be sufficiently fast enough to allow for the cell to survive. So 1 in 10,000 is a high enough quality control or error rate, or low enough error rate, I should say, that you can more than likely make all the proteins that you're going to need in the cell with a high degree of fidelity, and yet fast enough that the cell is able to actually operate. And even then, there are mechanisms in place that if a protein is made incorrectly, it is tagged for destruction and
degraded
and be destroyed by the proteasome. So there's an additional quality control checkpoint at the very end that helps to compensate for even a 90% error, production process of production.
And we're looking at the 1,000 amino acid protein, which is a very big one. So if we were looking at a smaller one,
right,
then 1 in 10,000 will give a huge, a very good, much better than 90% fidelity, right?
Yes,
and
exactly.
I'm just trying to connect the dots here. So again, it makes very good sense that the proteins that make the ribosomes themselves are small proteins compared to, they are not 1,000 amino acid proteins. They're the smaller ones, which at 1 in 10,000 error rate would give maybe, 99 point something % of fidelity. So the components of the assembly line machines themselves are produced with the optimum small size, not the very small size, but the optimum small size that they will be themselves produced at very high fidelity. Did I get that connecting the dots right?
Oh yeah, that's exactly right. That's another reason why, again, you want again smaller than typical, right? Smaller than typical, because if you have a protein of about 300 amino acids, is a pretty typical protein size. And there you're pushing very close to probably 99%, accuracy. And so if you're going smaller than that, then you're almost guaranteed that the ribosomal, proteins are going to be made correctly.
And maybe this is also an embedded wisdom that many of the machines and many of the protein complexes that we see are actually protein complexes rather than just one big protein folding. They're always those components coming together, and this reduces the problems per component. So we have high fidelity components that then bind,
right.
And then probably if you have one of the components defective, it will not bind. So this will on its own form another protection system, because if it's not the proper amino acid peptide chain sequence, then it will probably not fold properly. That will probably not fit in its place of the protein complex anyway. So it gives this second layer of, if the Lego piece is not the right Lego piece, if it's defective, it will not fit in the bigger assembly of Legos, right?
Yes, that's right. That's right. Yeah, and that's incredibly insightful on your part. But to me, again, being around a manufacturing operation or several operations for 10 years, you come to appreciate the ingenuity in quality control. Good quality control systems are as much an engineering marvel as the actual design and product and the manufacturing operation itself. And so to see that level of quality control and the elegance of that type of quality control connected to protein manufacture, to me, is really impressive. In and of itself, that to me is enough to convince me that this is all designed, right?
Yeah, absolutely. Actually, everything that we have gone through this series, Fuzz, is immediately lends itself that this is not just design. This is design genius. This is unbelievable level of design on very, 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 very nano scale, and this is essentially, we're just scratching the surface of nano engineering and nanobots, etc. And this is operating at molecular level with all this sophistication and error detection and error correction, and then quality control and quality assurance, and then even after anything passes, there is those gatekeepers that prevent them from spoiling the whole system. And I think even at more macro level, if the cell finds that there is too much problems with it, it might even commit suicide and initiate those other crazy mechanisms like programmed cell death and those other things. And this whole thing is not, it doesn't seem to me, actually, I'm sure this is not a system that is built to appreciate and welcome random mutations to come and shape it. It doesn't look like this at all. And still there are some people who would just like look the other way and refuse to appreciate the marvel of our creator in his creation. Did we really finish translation here? Because I will really miss Central Dogma.
Yeah, I think we've run our course here. But we're going to talk in the next episode about protein folding, which is really now looking at it. Technically is not part of the Central Dogma, but it really is looking at, now that you've made that protein chain in a single dimension, its real utility is in its three-dimensional structure. And so how do proteins fold? And so we'll offer some thoughts about that as well.
But one insight that we might think of here is that this whole system has just produced this chain of beads, and this specific chain of beads will take a specific 3D shape that is only possible through this chain of beads. So the system also has this foresight that I want to produce this 3D shape through this chain, as if it knows the result of protein folding. And it occurs that protein folding is one of those crazy engineering problems that we are just starting to crack the surface. What will be the corresponding 3D shape of that peptide chain? And only very sophisticated AI can do it these days, I guess. I think only this last year that we're scratching the surface. So this will be quite an exciting episode, and I think it will have some of those paradoxes too, right?
Yes. Yep. And what we'll see is there is design in a surprising way in the process of protein folding. The system seems to be jimmy rigged from the very get-go.
I think everything in this system is like It's like that. So everyone, this was really coronating our central dogma installments, and please give us your questions in the comments, and see you on the other side for the after show. Fuzz, this has been, as usual, another exciting session of yours. So thank you for the teaching, and thank you for helping us see God's marvel in His creation. And see you on the other side for the after show.
Yeah.